हेलो फ्रेंड्स, स्वागत है आपका एक और वीडियो में। हियर वी आर गोइंग टू डिस्कस द एमएससी बायोकेमिस्ट्री ड्यू 2021 पेपर. So let's begin. The scientist who identified the transforming agents in Griffith's famous experiment as DNA was Irving Chauger, Oswald Avery, Frederick Michel, or Peter Michel. In 1944, Oswald Avery, MacLeod McCarty, and Colin MacLeod they identified the transforming agent in Griffith experiment was actually DNA. So here the option B has to be the correct answer. The scientist who created the first recombinant DNA molecule was Howard Temin, Paul Berg, Siemens Shapiro, or Ian Wilmot. In 1972, Paul Berg made the first recombinant DNA molecule using ligase, while in the same year, Stanley Cohen, Annie Cheng, and Herbert Boyer produced the first recombinant DNA organism. So, option B has to be the correct answer. Which of the following pair of amino acids are both glucogenic and ketogenic in nature? Alanine and lysine, lysine and leucine, isoleucine and phenylalanine, or aspartate and lysine. If you can see it in the figure, you can figure it out that lysine and leucine, they are the pure ketogenic amino acids, while there are 5 sets of amino acids which are both glucogenic and ketogenic. They are isoleucine, leucine, phenylalanine, tryptophan, and tyrosine. So for the given question, the answer has to be the option C, that isoleucine and phenylalanine are both glucogenic and ketogenic in nature. Next, the coenzymes in list 1 items serving as a transient carrier of specific atoms or function group in list 2. So list 1 is having 4 coenzymes and you have to find out that to which group or functional group it is going to react. So let's start. Coenzyme A. Coenzyme A is basically has a terminal cysteine residue or SH group to which an acyl group is tethered. Generally, coenzyme A is also known as acyl group carrier. So A has to be correctly matched with the 4 acyl groups. Now, FAD. FAD is often found as a redox cofactor and exists in 2 redox states. FAD, which is an oxidized state, and once it accepts 2 hydrogen atoms, it becomes FADH2, which is a reduced state. So FADB has to be connected with third. Now, pyridoxal phosphate. Pyridoxal phosphate is a coenzyme who is having a aldehydic group and react with the amino group of any amino acids or any other molecule which is having NH2 group and forms the Schiff linkage. So C has to be connected with 1. No. We have to find out to which group it reacts, not that the group it contains. So pyridoxal phosphate definitely has an aldehyde group, but it reacts with amino group. So C has to be connected with 2. Now, thymine pyrophosphate, it has a thiazole ring which is having an acidic proton and it can bind with the aldehyde group. So, D has to be correctly matched with the first aldehyde group because it reacts with aldehyde group. So, option A is connected with 4, B connected with third, C connected with second, and D connected with 1. That is option A. Glycogen phosphorylase exists in 2 forms in skeletal muscle. The active form of phosphorylase A is generated from phosphorylase B by reversible dimerization of phosphorylase B triggered by calcium ion, proteolytic cleavage of a decapeptide from the N-terminus of phosphorylase B, protonation of the active site histidine residue by a decrease in intracellular pH, or ATP-dependent phosphorylation of a specific serine residue on each subunit. In liver and muscles, glycogen phosphorylase enzyme is allosterically regulated through phosphorylation step. Active form of a phosphorylation type A is converted to inactive phosphorylation beta type by the reversible phosphorylation of a serine 14 residue. So here the option D has to be the correct answer, that ATP dependent phosphorylation of a specific serine residue on each subunit is basically responsible for the conversion of active form of a phosphorylase A from phosphorylase B. What are bile salts? Charged phospholipid, amphipathic cholesterol analog with detergent properties, esterified cholesterol, or hydrolyzed form of a triglycerol. Naturally occurring bile salts are derived from the cholesterol and are amphipathic detergent types molecule. In water, they form micelles which are required for fat emulsification. So option B has to be the correct answer. Given below are the events in the cell cycle. So there are 4 events which are being given and we have to arrange as per the correct order in the occurrence. Option A was phosphorylation of laminin A, B and C. B. Phosphorylation of retinoblastoma protein. Polyubiquitination of securin. Or D. Association of inner nuclear membrane protein and nuclear pore complex proteins with chromosomes. So we have to find out what is the correct order in a typical mammalian cell cycle. As we know that cell cycle event is tightly regulated by the cyclin and cyclin dependent kinases, which are definitely under controls. Retinoblastoma protein is a nuclear protein that undergoes in phosphorylation and dephosphorylation in concert with cell cycle. To begin cell cycle, retinoblastoma is phosphorylated, which now cannot bind to the E2F transcription factor. This transcription factor is required to begin the cell cycle. So, when the decision has to be made to begin the cell cycle, E2F must be free. So, retinoblastoma phosphorylation actually makes sure that E2F remain free for the beginning of cell cycle. That is the first step. Once cell cycle is decided to begin, then laminin has to be depolymerized. These architectural proteins remain associated with animal nuclear membrane, and phosphorylation of laminin based depolymerization on the nuclear envelope break down before mitosis get complete. So that has to be the second step. One of the most important steps during mitosis is the separation of sister chromatid to the opposite poles. In order to do so, cohesin, a protein which glue the sister chromatids, need to be destroyed. And this destruction is being done by an enzyme called separase. Separase generally remain inactive due to the another protein called securin. So basically securin need to be destroyed in order to active the separase, which will ultimately going to deglue the cohesin. Polyubiquitination degrades securin, and in chain of events ultimately cohesin is degraded and finally anaphase is complete. So the polyubiquitination of securin should be the third step, which is occurring somewhere nearby anaphase. Finally, inner nuclear membrane complex and nuclear pore complex proteins regain its original position with chromosomes and a fresh nucleus is formed in a daughter cell. So the D steps has to be the last. So what should be the correct order? That the B, phosphorylation of retinoblastoma cells, retinoblastoma protein, which will make sure that it will free the E2F so the cycle can begin. Second, the phosphorylation of laminin ABC so that the nuclear membrane can be dissolved. The C, polyubiquitination of securin, which will make sure that the cohesin can be dissolved and sister chromatid can separate to the opposite pole. And last, that the new nuclear membrane will be formed. So the correct order would be B, A, C, D, which is option D. So option D has to be the correct answer. Which of the following diseases are related to coronavirus? MERS, cholera, polio, or smallpox? You must be wondering in past 2-3 years that how come coronavirus has infected the humans, which was earlier supposed to be found in the bats. Definitely coronavirus is a zoonotic virus which is found in the animals and has a potential to affect the humans when they comes in contact. But you might not be aware that in 2012 a similar kind of a virus, which is also known as MERS virus, has been found in the Gulf countries, which is supposed to be infected the camel first and then from camels has been transmitted to the humans. So MERS is also a type of coronavirus which has already affected the humans in 2012. Balls and sticks in Rasmol are solid sphere and rods representing atoms and bonds respectively, cylinders or spiral ribbons to represent alpha helices and broad flat arrows to represent beta strands, line drawing representing bonds between the atoms, or solid spheres and rods representing bonds and atoms respectively. Rasmol ball and stick model are the 3D models used to represent biomolecular structures, where each atoms are represented with a solid ball line spheres and stick represent bonds connecting two atoms. So here the ball and stick in Rasmol's are the solid spheres and rods representing atoms and the connecting bond respectively. The acid hydrolysis of lysosomes do not digest the organelle itself because lysosomal milieu does not provide optimum pH. These enzymes are present as zymogens. Lysosomal membrane is heavily glycosylated, or ultimately the lysosomes are also degraded. Lysosomal enzymes cannot digest themselves because most of the membrane protein of lysosomal membranes are heavily glycosylated. So option C has to be the correct answer. Which chemical you will use for quantifying DNA? Densyl chloride, diphenylamine, orsinol, or ninhydrin. Diphenylamine is used for the quantitation of DNA, where under acidic conditions DNA react with diphenylamine dye and form bluish green complex. Now the color can be measured at 595 nanometer. Always remember that this method is non-specific and nowadays not is much used. So option B is the correct answer. Which of the following bonds inside a cell can be formed by the COOH group of cellular amino acids? Amide and carboxylic anhydride bonds, ether and ester bonds, amide and ether bonds, or ester and amide bonds. Each amino acid has two functionalities, amino group and COH group. There are certain reactions which are due to the COH group, that is like salt formation with alkalis, ester formation with alcohols, amide formation with amino group, and decarboxylation. While the reaction specific for NH2 groups are deamination reaction and reaction with ninhydrin chemical. So here the option D has to be the correct answer, that the reaction specific to COH group present in any amino acid would be ester formation with alcohols and amide formation with NH2 groups. A protein sample, when subjected to reducing SDS parts, displayed two bands of molecular weight 20 and 25 kD. When the same protein sample was subjected to non-reducing isoelectric focusing, a single band was observed. These observations are best explained with the following relevant reasoning. The sample either contains two proteins of different molecular weight and same net charge, or one protein with two subunits that differ in molecular weight but share the same net charge. The sample contains two proteins of different molecular weight and same net charge, or the sample is heterodimer with the subunit sharing the same net charge but differing in the molecular weight. The experiment had some error and the results cannot be explained. Now, to understand this, let's see the figure. If we consider that a protein having two subunits, which can be connected with a disulfide linkages, and in the next case, there are two proteins with having a molecular weight of 20 and 25 kD. If you will run this protein on reducing SDS part, in the first case, if the disulfide bond will be reduced, then the 20 kD and 25 kD will give you two bands. While in the next case, since there is no disulfide bond is present in the protein, then it will also give you the same two bands. Since the molecular weight is not much different, so we can assume that the two subunits or the part of the single protein connected with the disulfide bond has the similar charges. So on isoelectric focusing, both the protein will give you single band. it cannot be differentiated whether the protein is connected with disulfide linkage and have two subunits, or there are two different proteins. If you really want to differentiate, then you should run a non-reducing SDS patch. If you will run a non-reducing SDS patch, then the first case will yield you a 45 kD band, while the second case yield you two bands of 20 kD and 25 kD. So here the option A seems to be close to correct. The sample either contains two proteins of different molecular weight and same net charge, or one protein with two subunits that differ in molecular weight but share the same net charge. Uncouplers like DNP or thermochainin uncouple electron transport chain and oxidative phosphorylation by inhibiting cytochrome oxidase, blocking electron transfer, dissipating the proton gradient, or blocking ATP-ADP translocase. Uncouplers of oxidative phosphorylation actually disconnect the activity of electron transport chain and the ATP synthesis. Actually, they allow the reverse migration of the protons which have been generated by the electron transport chain activity. Now the protons are get back to the matrix, and now there is no proton gradient is being left. Since there is no proton gradient is there, so no driving force is there to synthesize the ATP, and hence ATP synthesis is being compromised. So here the uncouplers like DNP or thermochainin basically uncouple by dissipating the proton gradient. An enzyme E has a kcat value of 600 per second. Now we have to calculate the Km value when reaction proceeds at a rate of 9.6 micromolar per second and substrate concentration is 40 micromolar. The total given concentration of enzyme is 0.02 micromolar. As we know, the formula of kcat is Vmax by total enzyme concentration. So if we place the value of kcat and total enzyme concentration which is being given, then we can get the value of Vmax. Now if we place the value of Vmax in a classical Michaelis-Menten equation, then we know that we have the values of V, that is instantaneous velocity, Vmax, S, and we have to find out the value of Km. Km, after solving the values, then we can get the value as 10 micromolar. So option C has to be the correct answer. Which one of the following is not a heme protein? Catalase, tryptophan pyrrolase, delta amino levulinic acid synthetase, or cytochrome oxidase? Heme containing proteins, they contain a special chemical called heme group which confer them with special functionalities like oxygen carrying capacity, oxygen reduction, or even electron transfer capacities. Heme is synthesized in bone marrow or liver with the help of delta amino levulinic acid synthetase. Hence, the third enzyme actually synthesize heme rather than contain heme. So, option C is the correct answer. Multi-enzyme systems include fatty acyl synthetases, hexokinases, phosphatases, or lysosomes. Fatty acyl CoA synthetase is a multi-subunit enzyme which catalyzes the ATP-dependent formation of fatty acyl CoA from free fatty acid. This step actually prepares fatty acid to yield ATP by using beta oxidation process. There is another enzyme which actually associated with fatty acid anabolism, that is fatty acid synthase. This enzyme is also a multi-subunit enzyme having 6 enzyme activities. So here the enzyme fatty acyl synthetase is an answer that it is a multi-subunit enzyme which is involved with fatty acid catabolism. The pi or isoelectric point of a protein is 6. When the electric field is applied at pH 8, the protein moves towards anode, moves towards cathode, does not move, or moves in random fashion. As we know that at isoelectric point, that is here, which is pH 6, the protein will be having equal number of positive and negative charge. That means it will be electrically neutral. When you keep any protein at very low pH, the protein develops the net positive charge. While if you keep any protein at an alkaline pH, they develop the net negative charge. So this protein whose pI is 6, if you keep it at a pH 8, then it is bound to develop a net negative charge. So definitely it will be attracted towards the positive electrode, that means anode. So the protein at pH 8 will be moving towards the anode. In glycolysis, pyruvate dehydrogenase is subjected to feedback inhibition by its products. The chemical compounds which might be involved in the process are listed below. A. NADH, B. FAD, C. acetyl-CoA and D. acetaldehyde. Now, which one of the following combination of above chemical compounds is involved in the feedback inhibition of pyruvate dehydrogenase? As you can see in the figure, pyruvate dehydrogenase can positively regulate by pyruvate and negatively regulated by NADH, acetyl-CoA and ATP. So here option D, that means A, which is NADH, and C, which is acetyl-CoA, will be the one which is going to inhibit the pyruvate dehydrogenase through feedback inhibition. The approximate molecular weight in kDa of the product after the translation of a 390 basis mRNA will be 48 kd, 26 kd, 39 kd or 40 kd. As we know that the number of base pair is given as 390 and each codon is having 3 base pair. That means total codon would be around 130. 390 divided by 3. Now an individual amino acid has a molecular weight of 110 dalton. So a peptide of having 130 amino acid will give me 14300 dalton or 14.3 kilodalton protein. So option D has to be the correct answer. Natural proteins are composed of 20 alpha amino acids. Which one of the following statements is true for any of these amino acids in a solution of pH 1.5? Only amino group is ionized, only carboxylic group is ionized, both amino and carboxylic groups are ionized, or both the amino and carboxylic groups are neutral. Always remember, every amino acid generally develops a net positive charge when it is kept at acidic pH, where amino group is protonated and net negative charge at high pH because the carboxylic groups gets deprotonated. So here the option A has to be the correct answer, that whenever any amino acid is kept at a very low pH, then only amino group is ionized. The arrangement of nucleotides in DNA can be seen by ultracentrifugation, X-ray crystallography, light microscope or electron microscope. Nucleotide arrangement in DNA can be seen by the X-ray crystallography, in which incident X-ray beams passes through the crystalline structure and gets diffracted into many specific directions. Watson and Crick proposed double helical structures of DNA based on the classical X-ray diffraction photo obtained by the Rosalind Franklin, which is also known as photo 51. So here the X-ray crystallography is the method of choice which can be used to see the arrangement of nucleotides in DNA. Amino acid residues which are predominantly involved in the protein DNA interactions are alanine, negatively charged, prolines or positively charged. See, DNA is negatively charged due to the phosphate backbone. So proteins interacting with the DNA must have positively charged or basic amino acid like lysine, arginine or histidine. So the amino acids has to be the positively charged as present in even histones. So D has to be the correct answer. Select the amino acid which can exist as a diastereomer: valine, proline, glycine or isoleucine. Stereoisomers that are non-superimposable, non-mirror image are known as diastereomers, and generally this phenomena is observed when a chemical have more than one chiral center. Isoleucine and threonine has got two chiral centers while glycine has none. So isoleucine and threonine will be existing as more than one diastereomers, while glycine will never produce a optically active compound. All the below given amino acids are optically active except glycine, phenylalanine, valine and tryptophan. As we have just discussed that glycine do not have any asymmetric carbon, hence it has to be optically inactive. So option A has to be the correct. Select the true statement about phosphodiester linkage. 3' phosphate group of one nucleotide unit is joined to the 5' hydroxyl group of the next nucleotide. 5' phosphate group of one nucleotide unit is joined to the 3' hydroxyl group of the next nucleotide. 5' phosphate group of one nucleotide unit is joined to the 5' hydroxyl of the next nucleotide, or 3' phosphate of one nucleotide unit is joined to the 3' hydroxyl of the next nucleotide. Phosphodiester linkage is created when the 5' end of one nucleotide having phosphate group react with the 3' hydroxyl of another nucleotide which is a part of the existing strand. So here the option B has to be the correct answer. Lecithin are composed of glycerol plus fatty acid plus phosphoric acid plus serine, glycerol plus fatty acid plus phosphoric acid plus ethanolamine, glycerol plus fatty acid plus phosphoric acid plus choline, or glycerol plus fatty acid plus phosphoric acid plus betaine. Lecithin are the phosphoglycerides made up of condensation of glycerol with at least one phosphorylated fatty acid, that is phosphoric acid plus choline and two fatty acids. So here the option C has to be the correct answer. Molecules that pass through a cell membrane most easily are small and hydrophobic, large and polar, ions or monosaccharides. Small molecules that are hydrophobic or non-polar, that means have no charge, can cross the membrane easily through the cell membrane, while ions like charged ones and larger hydrophilic molecules cannot pass through. So here the option A has to be the correct answer, that small and hydrophobic molecules that pass through a cell membrane most easily. Which of the following molecules can be utilized for establishing early evolutionary processes? Ribosomal RNA, mitochondrial DNA, chloroplast, or nuclear DNA. 16S ribosomal RNA gene is a universal marker for phylogenetic reconstruction because of its high conservation. So, ribosomal RNA is a established marker for establishing the early evolutionary processes. The enzyme responsible for conversion of negatively supercoiled DNA into a relaxed circular DNA is DNA gyrase, DNA topoisomerase 1, DNA topo 2, or DNA ligase. Generally, topoisomerase 1 introduces a single-stranded break in duplex DNA and remove supercoiling to the relaxed state. Now, topo 2, which is also known as DNA gyrase, is responsible for converting bacterial circular DNA to the negatively supercoiled DNA. That means what? Topo 1 actually releases negative supercoiling to make a bacterial circular DNA as a relaxed DNA. While Topo 2, or also known as DNA gyrase, it actually introduces negative supercoiling. So here the option B has to be the correct answer. Which of the following is not served by the plasma proteins? Blood clotting, oxygen transport, hormone binding and transport, or buffering capacity of the blood. Transport of oxygen is a function of hemoglobin, which is absent in plasma, rather present in RBC. So, oxygen transport cannot be performed by the plasma protein, rather by RBC protein. Only one X chromosome in the aneuploidy females is a characteristic of individual with Cry-to-Cat syndrome, Klinefelter syndrome, Down syndrome, or Turner syndrome. Sex chromosome aneuploids comprises loss or gain of sex chromosome, that is X chromosome. So generally all aneuploid females, they generally have either a higher copy of the sex chromosome X chromosome, or there is a loss of a sex chromosome, that means X chromosome. Turner syndrome is a loss of X chromosome, while Klinefelter is a gain of X syndrome. So in the question we have been asked that only one X chromosome is there. That means out of a two X chromosome, one is lost, which is a characteristic of a Turner syndrome. Which of the following signaling molecule does not interact with cell surface receptor? Insulin, gastrin, glucagon, or testosterone? That's very easy. Insulin, gastrin, and glucagon are peptide hormones which actually generally interact with the cell surface receptor as they cannot cross the membrane. While on the other hand, testosterone, which is an steroidal hormone, do not bind to the surface protein and cross the plasma membrane to interact with the DNA. So here the option D has to be the correct answer. Coenzymes involved in the formation of acetyl-CoA from pyruvates are thymine pyrophosphate, lipoic acid and FAD, pyridoxal phosphate, biotin and FAD, vitamin B12, folic acid and vitamin C, NADH, lipoic acid and vitamin E. During the end stage of glycolysis, pyruvate is converted to acetyl-CoA by an enzyme called pyruvate dehydrogenase complex. This complex requires coenzyme ASH, NAD as a substrate, and TPP, lipoate, and FAD as a coenzyme in sequential manner. So here the option A has to be the correct answer, that thymine pyrophosphate, lipoic acid, and FAD are the common coenzymes which are used by the pyruvate dehydrogenase complex along with CoASH and NAD. There are various subclasses of antibodies found in the body fluids and body secretions. Many different functions may be attributed to these subclasses. Given below in list 1 is major function of different subclasses and list 2 consists of the name of the subclasses. So there are various functions which are being given in list 1 item, while we have to correlate it what is the major function played by the which antibody. So now list 1 first item would be binds to macrophages by Fc. Macrophages and monocytes possess a surface receptor which can bind to the Fc region of IgG and plays its role in phagocytosis. So A list item has to be connected with the 4 IgG. Now next is binds to mast cells by basophils. IgE binds to the mast cell in tissues and basophils in circulations and generally involved in the allergic reaction. So B component has to interact with the third, that is IgE. Now C is first B cell receptor. IgM is the first B cell receptor as well as the first antibody molecule present in newborns. So C has to be connected with fifth, that is IgM. Now D is no major specific function known other than antigen binding. Definitely it has to be IgD because IgD is not known to have a specific function and it is debatable that why it is present in immune system. So D is connected with the 2. Now protection of mucous membrane. So IgA is an antibody which are associated with mucosal membrane. So E has to be connected with first. So if we see, then A is connected with fourth, B is connected with third, C is connected with fifth, D is connected with second, and E is connected with first, which is option D. Receptors present on macrophages can bind to. We have just discussed that macrophage possesses receptor for the Fc region of the IgG, which is option A. Match the techniques in list 1 with the application given in the list 2. So list 1 outlines the different techniques, while list 2 outlines their basically specific uses. A. Salting out. B. Ultracentrifugation. C. Dialysis. While D. Isoelectric focusing. See, salting out is a method of protein precipitation at high salt concentration. That means A has to be connected with 2, protein precipitation. While dialysis, which is C, is a method which is used to remove these salts from protein precipitation. That means C has to be connected with 4, removal of low molecular weight, that means salt impurities. So we have to find out in which option A is connected with second while C is connected with fourth. If you see the option A is connected with second while C is connected with fourth, that has to be the correct answer. Ultrafilament centrifugation, as we know, is used to determine the sedimentation coefficient, while isoelectric focusing is also used to determine the PI. 5' end capping of mRNA transcripts in eukaryotes involve the following events. A. Addition of GMP on the 5' end. B. Removal of gamma phosphate of the triphosphate on the first base at the 5' end. 5 to 5' linkage between GMP and the first base at the 5' end. Or D. Addition of the methyl group to N7 position of guanine. Now we have to arrange in a correct sequence. If you see it in the figure, that the steps of 5' cappings are: first, gamma phosphate removal at the 5' end. Second, the addition of GMP at the 5' end. Third, condensation of GMP with beta phosphate at the 5' end. Now last, the addition of methyl group at the N7 of guanine. If you correctly sequentially arrange these, then the B steps come to the first, A steps come second, C steps come third, and D steps come last. So B, A, C, D has to be the correct option, that means option C. Arrange the following events of meiosis in correct sequence: crossing over, synapses, terminalization of chiasmata, or disappearance of nucleolus. Steps will be chromatin condensation, which happens to be in leptotin; synapses, which happens to be in the zygote; crossing over, which happens to be in the pechite; chiasmata appearance, which has to be in the diplote; while chiasmata terminalization followed by nucleolus disappearance, that has to be in the dikinases. So the option has to be B, synapses first; A, crossing over second; C, terminalization of chiasmata third; D, disappearance of the nucleolus, that is D. So option BACD is the correct sequence, that is option B. Identify the correct name of a protein present only in the biological membrane: influenza, hemagglutinin, BSA, insulin, or trypsin. Influenza hemagglutinin, HA, is a homotrimeric membrane glycoprotein found on the surface of the influenza flu virus, and it is responsible for its infection along with an enzyme called neuraminidase. So here the option A has to be the correct answer. The protein involved in photosynthesis is hemoglobin, collagen, rubisco, or albumin. Rubisco or ribulose biphosphate carboxylase is a universally present enzyme in all photosynthetic plants. This is very simple lower class question which you can remember. So option C has to be the correct answer. Sialic acid is a part of phosphatidylcholine, ganglioside, cholesterol, or triglycerides. Sialic acid are carbohydrate molecules with a C9 carbon backbone and a carboxylic acid group, which is commonly present as a part of glycoprotein, glycolipids, and gangliosides. So option B is the correct answer. Photon capturing moiety of chlorophyll molecule is known as porphyrin, magnesium porphyrin, chlorophyll B, or chlorophyll A. In chlorophyll, magnesium porphyrin is the moiety which functions to capture energy and store electron during photosynthesis. So option B has to be the correct answer. Humoral immune response in higher animals is involved with B cells, T cells, both B and T cells, or macrophages. See, it's a very confusing question. Whenever we hear humoral word, B cells is automatically comes in our mind. But remember, specific humoral activity actually involves the action of B cells or antibodies under the guidance of T helper cells. So answer has to be the both B and T cells. Which one below can break a peptide bond enzymatically? Papain, myosin, cyanogen bromide or performic acid. Papain is a protease which can break peptide bond by using its catalytic triad with a deprotonated cysteine at its active site. During UV radiation of DNA, the following compound is formed. That's very easy. Thymine dimer is a photolysine which is produced due to the exposure of UV radiation to a DNA. Hence, option A has to be the correct answer. Protein is degraded inside living cells through a key process of farnesylation, ubiquitination, methylation or palmitoylation. Ubiquitination is a process in which a protein subjected to degradation is tethered with a ubiquitin sequence and recognized and degraded by the proteasome. So option B is the correct answer. The observation that plasma membrane protein mix after cell fusion provide evidence for rotational movement of plasma membrane proteins, the bilayer structure of the biomolecules, the fluidic mosaic model, or interaction of plasma membrane proteins of two different cell types. An experiment performed by the Fry and Adidin provided evidence in support of the membrane fluidity and dynamism. First, you need to understand what was the previous model. Davidson and Daniele proposed a tri-layer model of the lipid bimembrane, which suggests that the lipid membrane used to be sandwiched between two protein layers. Now, when two cells will fuse, as per their model, the protein layer must get locked between two lipid bimembrane layers. But as far as Fry and Adidin reports were concerned, they found that the protein of both the layers they get diffused and mingled, means that the layers are fluidic in nature and can mix. So option C is the correct answer, that the Fry and Adidin model suggested that the lipid bilayer are fluidic in nature rather than static. Several classes of hydrolases are localized in. Again, one of the most simple question, that in the lysosomes, there are various types of hydrolases are present to degrade various biomolecules category, including proteases, lipases, nucleases, or even glucosidases. Which cofactor is crucial in catalyzing enzymatic conversion of pyruvate to lactate? NAD, NADH, FAD, or FADH. If you see it in a diagram, that lactate dehydrogenase or LDH, which catalyze the conversion of pyruvate to lactate using NADS as a cofactor. So option B is the correct answer. The chemical nature of covalent linkage in sucrose is known as N-glycoside, O-glycoside bond, amide, or diester. Sucrose is composed of alpha-D-glucose condensed with beta-D-fructose connected with alpha-1,2 linkage, which is a O-glycosidic type bond. So option B is the correct answer. In sickle cell hemoglobin, which one out of the following is true? A glutamate is substituted by a valine, a glutamine is substituted by a valine is substituted by a glycine is substituted by a valine. This is again one of the most simplest question which we are encountering since our lower classes. In sickle cell hemoglobin, a glutamate, which is a charged amino acid, is replaced by a hydrophobic long chain valine amino acids. So option A has to be the correct answer. With this, we sum up our video. If you like our content, please do subscribe in large number and comment, like and share. Thank you very much.